Hello, everyone. Thanks for attending this symposium on neuropsychiatric genetic variation, shaping brain architecture by modulating gene expression. My name is Sebastien Jacquemont. I'm a medical geneticist here in Montreal at the Pediatric Hospital of Saint-Justine, and my research focuses on understanding the effect of psychiatric mutations on cognition, behavior, brain endophenotypes, and risk for disease. So we're interested in understanding these mutations because, first of all, psychiatric conditions are highly heritable, and the genetic contribution ranges anywhere from 40 to 80%. What people might not know is that beyond the research, genomic testing is widely implemented in the clinic, and in a pediatric hospital like ours, thousands of tests are ran annually, and about 10 to 30% of children with developmental or psychiatric conditions such as autism or learning disabilities carry high-risk mutations. On the other hand, even though this is widely used in the clinic, little is known on the effect of these mutations on brain endophenotypes. I'm just going to give you a few examples of what this research has taught us in the past 5 to 10 years. And the first example is on a simple trait such as total cortical surface area. And what I'm showing here is that if you modulate gene expression on chromosome 1, a small region of chromosome 1 that contains about 5 to 6 genes, if you decrease expression at that region, total surface cortical area will decrease by and the effect size is quite large, beyond 1 standard deviation. If you increase expression of the same genes, you will increase total cortical surface area. If you look at another region on chromosome 16, you have the same phenomenon, except that it's swapped around. Decrease in gene expression at that region, which contains about 30 genes, will increase total cortical surface area, and vice versa. There's more data coming out right now suggesting that this is not specific to these regions, and you see it in other regions of the genome. You can look at other brain endophenotypes, and Clara Moreau will discuss the effect of mutations on functional connectivity. Here I'm giving an example of what you observe in people who carry deletions. So deletions lead to a decrease in gene expression. What you observed in people who carry different types of deletions is a particular pattern of disconnectivity characterized by an increased connectivity of the thalamus and the basal ganglia, the somatomotor network, and a decrease in connectivity in the visual and frontoparietal networks. So there's clearly very robust effects of these mutations on the general brain architecture, but mechanisms by which these associations occur remain mostly unknown, and this will be the topic of our symposium. The first speaker will be Paul Thompson, a professor at the University of Southern California, and he will discuss genetic determinants of brain structure using data generated by the Enigma Consortium. The second speaker will be Clara Moreau, who is a PhD candidate here at the University of Montreal, and she will discuss the effect of high-risk mutations for psychiatric conditions and their effect on functional brain networks. Jason Stein is an assistant professor at the University of Northern Carolina, and he will present data on molecular mechanisms by which common genetic variants lead to changes in brain structure. And finally, Trygve Bakken, who is an assistant investigator at the Allen Brain Institute, We'll discuss molecular diversity of cell types in human cortex, the neuronal specialization across brain areas, and homologies across species. So without any further ado, we will start with our first speaker, Paul Thompson.
Hi, so the title of today's talk is Genetic Determinants of Brain Structure, and my name is Paul Thompson, and I'll be speaking on behalf of the Enigma Consortium. So I'll give you a quick overview of the talk. It'll be about 10 minutes. I'm going to first describe Enigma, a worldwide alliance to map disease and genetic effects on the brain. I'll also explain some of the methods for genome-wide association studies of brain measures, and also review some of the more recently discovered findings of genetic influences on brain structure, function, and rates of brain change in longitudinal data. And then at the end, I'll tell you a little bit about the newer multivariate methods that can be used to detect gene effects on the brain. So, a little over 10 years ago, Enigma began a very, very large scale international analysis of what has now become over 100,000 brain scans. And there were two main goals. One is to understand how do 33 major disorders affect the brain, as seen on MRI scans or with diffusion tensor imaging, resting state functional MRI or EEG. And the second goal was to understand how genetic variation affects the brain. So we screen in Enigma millions of markers across the genome. If it's a genome-wide association study, these are single nucleotide polymorphisms, it's methylation levels in epigenetic analysis, or it may also be rare variants, as you've heard other speakers talk about copy number variants that are either expansions or deletions in the genome. And the overall interest is to find genetic markers and other factors that affect brain development and aging, or disease risk, or treatment response. So here's where the data comes from. The images and genetic data are collected all over the world. The colors denote the disease or disorder that the group involved mainly studies. But most of the studies I'll tell you about are in healthy controls. You can also do them in patients as well, but the majority of this work is done in healthy controls to look at normal variation. So here's how it works. So in a genome-wide association study, you can measure a certain phenotype from brain scans, whether the size of the brain or the microstructure of the corpus callosum, or it might be the activation of part of the brain. And then in each subject, you march along the genome, and this is imputed data that only includes the common genetic variants. And you ask the question using a regression, is there any evidence that variation at that locus in the genome is associated with differences in the volume or the microstructure or the function of the measure of interest? And it's standard regression after adjusting for other confounding variables. So when you do this, and this is a paper led by Derek Hybar in Nature, you get Manhattan plots or plots of the evidence that different locations in the genome, there you see the chromosomes on the x-axis, are associated with, in this case, the volume of subcortical structures. And for each structure, the hits or the genome-wide significant loci are a little bit different. So how is this done? So practically, we ask sites all over the world to compute brain measures from their MRI scans or DTI scans. Maybe they run FreeSurfer for morphometry. They might run TBSS, part of the FSL toolkit for DTI. And then separately, they all run genome-wide association measures that test evidence for association between a little over 1 million markers in the genome and the brain measures. And then centrally, we meta-analyze the effects from all the sites. So each site's vote depends on their sample size, and this makes sure that the effects are reproducible and can boost your power to pick up effects that no site can pick up on its own. So when you do this, and this is a study of genetics of hippocampal volume in data from 65 scanning sites across the world, you can begin to discover genetic loci associated with a volume, in this case of the hippocampus. Because we're brain mappers, you can map where these polymorphisms in the genome affect the brain using statistical parametric mapping. And you can even do some iteration to see genetic modules and where these genes have greatest effect. And the genes don't always exert their effects uniformly across life. So in this example, we see that the ApoE4 haplotype, which if you carry it triples your risk for Alzheimer's disease, that's something where later in life, at the age of 60 to 80, it shifts your hippocampal volume on average by 2 standard deviations away from the volume of people that don't carry this haplotype. But when you're younger, when you're maybe 20 or in your early adult life, it has no effect at all. So you can begin to see if these dangerous risk genes for disease are operating in the brain at different points in life. Now, this has been expanded. So Enigma teamed up with the Charge Consortium and the UK Biobank to do genome-wide association of volumes of all these subcortical structures in a little under 40,000 people. you can see a huge array of hits that are generally different for different structures. If you do this for the cortex, this is a new paper recently in Science by Katrina Grasby and Neda Jahanshad and colleagues. Now in a little over 50,000 people, you can see individual loci that are associated with the surface area or the thickness of different parts of the brain, again, partially overlapping, partially distinct. And this ideogram shows a huge survey of the genome with markers that have been associated with, in this case, cortical measures. We now know that common genetic variation that happens in more than 1% of us, accounts for over a third of the variation in the total surface area of the cortex and over a quarter of the variation in the average thickness of the cortex. There are some hotspots. So here on chromosome 15, there's a huge number of hits. These areas of the genome have variants that influence a large number of traits in the cortex, the surface area of different areas, the thickness of one area. And you can also ask if any of these variants that influence the size or geometry of the brain are implicated in psychiatric or neurological illness, and they are. So we know that the loci that tend to reduce surface area significantly overlap with loci that confer risk for major depressive disorder, ADHD, insomnia. We also know that conversely, genetic loci that tend to expand the cortex are associated with better cognitive performance on cognitive tests. And one conundrum is we don't know why, but it appears that Parkinson's disease is more prevalent in people that have genetic loci that tend to expand the cortex. We think this might have something to do with the MAPT gene, a locus that's been implicated in Parkinson's and also in the expansion of the cerebral cortex. So is this just structure or is it function as well? Well, you can measure brain function with a variety of methods here with EEG, and you could do the same thing. So Dirk Smit, leading Enigma EEG, has performed a genome-wide association study of measures looking at the power spectrum of functional synchrony with EEG. And in this study in human brain mapping a couple of years ago, he and his colleagues around the world essentially found that the GABA2 gene, which is involved in GABA-A inhibitory neurotransmission, affects beta power in the brain. This was a gene that was already implicated in alcoholism and epilepsy, but it was unclear why. But now it looks like this marker might affect the functional synchrony of the brain. So we're beginning to really carve out some exciting links between genetics, functional synchrony, and disease risk. And there's a little bit of a survey here in a new paper by Dirk Smit and his colleagues on PsyArchive now. Another interesting variant is you can look at the rate of change of brain volume. As we know in development, if you scan people repeatedly, there's very rapid growth, and there's thinning of the cortex, and then there's atrophy late in life. So you can look at, are there markers in the genome that affect the speed of development or the rate of degeneration of the brain? And there are. So Rachel Brouwer and Hilke Hulshoff-Paul and the Enigma Plasticity Group essentially combined data from 37 worldwide cohorts. In this case, people scanned not once but twice with an interval on average of 3.5 years. And they assembled a database of 10,000 people who'd been scanned twice. And these plots for hippocampus and ventricular volume show that in the early part of life, there's not a lot of change for the hippocampus, and then there's a precipitous decline. And then each dot here is a cohort, and the size of the dot is dependent on the size of the cohort. For ventricular volume, there's sort of a precipitous increase in volume starting at age 40, but then very much so after age 60. So when you do this for every structure that you can measure, you see this sort of very interesting rate of change graph for each structure, some of them plateauing and then precipitously declining in old age, others doing something a little bit different where they decline early in life and then plateau. But then they proceeded to a genome-wide association aggregating data from 10,000 people. And they've already found 15 genes that are implicated in the speed of development, the rate of growth of volume of parts of the brain. And this is a very interesting thing for clinical and translational use. So you could essentially imagine a polygenic score that you could compute that predicts to some extent how fast the brain would develop or how fast the brain would age. And then you could overlap that information with other aggregated information on different types of conditions. But can we be more efficient? So in all the studies so far, we've been looking at single point loci in the genome and single measures in the image. We know that in general decoding efforts here in the Enigma machine, which was used for code breaking during the Second World War, there were very sophisticated methods used to crack it. And just to give you a hint of what those methods might be in imaging genetics, There are certainly methods, in fact, here's one pioneered by Marco Lorenzi and colleagues in PNAS, where you take an aggregate of loci in the genome and an aggregate of features in the image, and you try and maximize the explanatory value of the gene set in explaining the patterns that you see in the image. And you can do this with canonical covariates analysis, sparse CCA and all of its variants. Another way to do it is with machine learning. So this review by Li Shen and myself in the proceedings of the IEEE talks about all the ways you can select sets of genomic markers and sets of phenotypes in images, they may be even multimodal images, and maximize the explained variance while also satisfying other conditions. You might want to pick genes that lie in a certain known pathway, or you may want to encourage sparsity or some other characteristic of the selection. So other new directions involve the analysis of whole genome sequences, which we're just beginning to do. Enigma also has working groups on epigenetics, looking at methylation and its influence on volumes of some polygon structures. And also, as you've already heard in the symposium, Enigma has efforts to look at copy number variation, these very large-scale deletions or duplications that often have much more dramatic effects on the brain than the single SNPs that we're talking about here. Finally, there's a new project to look at genome-wide association of DTI measures, and you're very welcome to join in if you want to. Go to this website and join in. We can either give you information on how to participate or just give you updates on the project. So just to conclude, we really want to acknowledge all of the people around the world. In fact, there's over 2,000 scientists who've contributed to Enigma, either genetic or disease studies from over 500 institutions in 45 countries, and also want to acknowledge the funding. It's very, very wide international support. Just want to say particularly thank you to the NIH for funding the Enigma groups that you see here, and to all of the patients and participants throughout the world who've made this project a success. So thanks very much for listening. We really appreciate you being with us today. Bye-bye for now.
So hi everyone. So I will present results obtained by studying mutation effect on connectivity and the relation to idiopathic autism and schizophrenia. So autism spectrum disorder and schizophrenia have been extensively studied through a top-down approach, starting from the behavioral diagnosis and moving down to brain intermediate phenotype and genetic factors. This biomarker race at the imaging level provided reproducible signature per condition. This also revealed shared intermediate phenotype across conditions, such as a widespread decrease of connectivity in ASD and schizophrenia and the thalamocortical dysfunction. This finding could be related to the polygenic architecture of this psychiatric condition, with several hundreds of rare in blue and common in green variants involved in each condition independently, but also across them, which suggests pleiotropy mechanism. Pleiotropy happens when a mutation in the same gene leads to multiple seemingly unrelated diseases. Such results raise therefore questions about clinical boundaries used to perform top-down approach. By recruiting groups based on genetic risk factor for autism and schizophrenia, genetic first study provide an opportunity to track mechanisms related to biological risk regardless of any clinical symptoms. CNVs are deletion or duplication of a DNA segment that encompass typically several genes. They are typically reported in 10 to 15% in children seen in the neurodevelopmental clinic, and they have been associated to a wide range of conditions, including schizophrenia with different penetrance, ASD, and ADHD, reflecting again the pleiotropy. But to date, no study compared effect of this psychiatric genetic variant on functional network in humans. It's why we first attempt to characterize the connectivity signature of those recurrent CNVs to use them as proxy to dissect the heterogeneity of psychiatric condition with a bottom-up approach. We speculate that they may represent dimension contributing to idiopathic condition. For this purpose, we analyzed 6,635 subjects with resting state data from 9 consortia. CNV were distributed into 3 categories: high risk, medium, and neutral CNVs without association. We parcellated the brain into 64 functional regions and obtained 2080 correlation coefficient. Each connectome was then standardized based on the variance of the control group. So we first compared the effect size of the functional alteration observed in these CNVs. So this density plot represents the distribution of the 2080 beta values of each contrast. We reported that the effect size of CNVs on connectivity was correlated with the clinical risk conferred by mutation. In other words, effect size was increasing with the CNV severities. We then asked whether psychiatric cases compared to psychiatric control had a significantly higher or lower similarity to the group level functional connectivity signature of a CNV using a Mann-Whitney U test. We summarize this result into this three brain map. So for schizophrenia, ADHD, and ASD. And overall, we can observe higher similarity of deletion than duplication with psychiatric condition, higher similarity of CNVs with schizophrenia and autism, and that a core set of regions were recurrently involved in this similarity. So we summarize this set of regions in this final brain map. And you can see that the thalamus and the posterior cingulate exhibited the higher degree of similarity between all neuropsychiatric CNVs and individual with psychiatric condition. Such results raise questions about pleiotropic effect and suggest biological mechanism shared across these CNVs. Such mechanism may be reflected at the expression level. That's why we investigated the association between the expression profile of the gene encompassed in both loci that we previously remapped on the functional atlas 64 by 64 and the connectivity profile of both deletion using partial least square regression. Using two components, we showed that their expression explained respectively 24% for the 16p and 43% for the 22q of the variance of the connectivity profile. We then investigated if specific region had higher degree of variance explained. So overall, we identified that the highest one were the basal ganglia and the visual network for 16p and 22q. We then correlated the connectivity profile of both deletion with expression of the 15,000 genes across the genome with expression data available. And we investigated if specific genes encompassed within both loci could be related to the CNV connectivity signature. So that's the distribution of the gene. And overall, we showed that only 2 over the 29 16p 11.2 genes and 3 over the 50 22q genes were correlated to the connectivity architecture. We also showed that many other genes were significantly correlated to those profiles, in particular for the 22q. This suggests more general mechanism underlying these connectivity patterns. We therefore extended this study to any deletion with the hypothesis that entire classes of genetic mutation may reorganize connectivity along general dimension irrespective of where deletion occur in the genome. In other words, all deletions may share a common connectivity pattern. For this purpose, we used all CNV available for analysis and annotated any deleted gene with an intolerance to loss of function score called PLI. So we computed the sum of PLI of all genes encompassed within each genomic loci. So for example, the 29 genes encompassed in the 16p1.2 CNVs per subject. So each carrier obtained a score corresponding to the gene content of his CNVs, and we investigated the linear effect of this score on connectivity. We identified for the first time that intolerance to haploinsufficiency was related to a connectivity pattern characterized by increased connectivity in the thalamus, the somatomotor, on the medial DMN, and decreased connectivity between the frontoparietal, the anterior DMN, and the lateral DMN. We summarize this pattern within this brain map that represents the contribution of each region to this pattern. So what it means is there is a common pattern across all deletion, and the effect size of the is the effect size of the pattern increased with the deletion severity. But then we ask if this pattern translates to any cognitive score or dimension. And we showed that individuals with lower general intelligence score showed similarities with 20 out of 64 regions of the haploinsufficiency pattern. This negative association was significant across 3 cohorts with general intelligence measure. And autism severity score also increased in individuals with higher similarity to the PLI pattern in 11 for the ADOS and 3 regions for the SRS and 3 regions. So overall, we showed that effect size of CNVs on connectivity is correlated with the known level of clinical risk conferred by mutation. We showed that neuropsychiatric CNV reorganized brain connectivity architecture along dominant patterns contributing to this complex idiopathic condition, in particular schizophrenia and autism. And finally, we identified that haploinsufficiency was associated with a general profile of disconnectivity, irrespective of where the deletion occurs in the genome. Such result could be seen at the transcript at the gene expression level. So I would like to thank all my colleagues, in particular Sebastien and Guillaume Huguet, my PhD supervisor, Sebastien Jacquemont and Pierre Bellec, and also to all our collaborators and the families, because none of this study could have been done without this worldwide collaboration. So thanks to everyone. Thanks for your attention.
Hi, everyone. My name is Jason Stein. I'm an assistant professor in the Department of Genetics and the Neuroscience Center at the University of North Carolina. The title of my talk today is From SNPs to Cortical Structure, Molecular Mechanisms by which Common Genetic Variation Leads to Changes in Brain Structure. So we formed the Enigma Consortium in 2009 to discover genetic variation impacting human brain structure. By pulling together brain imaging data from MRI and genetics from over 50 cohorts all over the world, we have had sufficient power to identify hundreds of genomic loci where variation is associated with brain structure. In this toy example shown here, the individuals with the T allele at SNP-A have an increase in brain structure. This is the type of association study that the Enigma Consortium has conducted. So though we've discovered hundreds of loci impacting brain structure, these loci are in general difficult to interpret. So I show here one example locus. So each dot represents a SNP, a single nucleotide polymorphism, where the location on the x-axis represents the location on the genome. The gene models are displayed below, the locations of the genes. And on the y-axis represents the significance of the association to a trait. In this case, the trait is total surface area of the cortex. So each of those dots above the line indicate a genome-wide significant association. So above this line indicate a genome-wide significant association. So this graph shows a genome-wide significant locus associated with cortical surface area. None of these significant dots are changing the protein coding sequence of the genome, though, and instead are found in non-coding regions of the genome, where we don't understand the functionality very well. Because DNA is inherited in chunks together during meiosis, genetic variants close to each other are correlated in a population. The correlation is called linkage disequilibrium, or LD, and is shown in the colors here. The more red color means a higher correlation. This makes it difficult to identify the causal variance within a locus. Also, though this locus overlaps the gene CENPW, we don't know if variation at this locus actually impacts this gene. And finally, the cell type in which this variant exerts its effect is unknown. Because most of the variants lie in the non-coding genome, we often think that they have gene regulatory potential. An overall model of how genetic variation in non-coding genome impacts brain structure is shown here. Our model is that first, a genetic variant can disrupt transcription factor binding in a non-coding gene regulatory element like an enhancer or promoter. This can lead to cell type specific changes in chromatin accessibility, which is an indirect measure of transcription factor binding or function of the non-coding genome. Subsequently, this can impact gene expression, again, in a cell type specific manner. Alterations in gene expression can then modulate cellular behavior that can have downstream impacts on brain structure. So this in part can explain some of the mechanisms by which genetic variation can impact cortical structure. The first step to understand that regulatory mechanism is to identify which cell type the variants are exerting their effect. In order to do this, we use an approach to determine if genetic variants associated with cortical surface area were enriched in regulatory elements present during certain time periods or within certain cell types. First, we found that there was a significant enrichment for cortical surface area heritability in fetal-specific brain regulatory elements, but not in adult-specific brain regulatory elements. This indicates that genetic variation impacts regulatory elements during fetal development to influence adult cortical surface area. Probing further and studying regulatory elements present within different cell types of the developing cortex, these cell types present here, we found the strongest enrichments for global surface area heritability within various subtypes of neural progenitor cells. This implies that genetic variation acts within human neural progenitor cells of the developing cortex to impact adult cortical surface area. Seeing that this time period and cell type was important, we then wanted to test how genetic variation impacts chromatin accessibility and gene expression during neurogenesis. We also wanted to perform this in a cell type specific assay because neural stem cell cultures are notoriously heterogeneous. So we took fetal cortical tissue from 92 donors in Dan Geschwind's lab at UCLA, where I was a postdoc. We isolated neural progenitor cells, cryopreserved them, and shipped them to UNC, where we performed the rest of the culture. At UNC, we grew up these cells and differentiated them into neurons in my lab. We labeled the neurons with a virus driven by a synapsin promoter, AAV2 HSyn1 EGFP. We then isolated the cells at 2 time points. The first time point is a progenitor time point. Neural progenitors are almost entirely, at this time point, are almost entirely a population of radioglia. They're SOX2 positive and PAX6 positive, which are markers of radioglia. Their differentiated neuronal progeny show typical neuronal morphology when labeled with this synapsin virus. At these 2 time points, we then performed chromatin accessibility assay using ATAC-seq. This is an assay where a transposase preferentially inserts into accessible or open regions of the chromatin. And we performed a gene expression assay, RNA-seq. In addition, all donor lines were genotyped so we could relate genetic variation to chromatin accessibility and gene expression. And in total, we had 92 donors in this study. Using this cell type data, we were able to discover thousands of places where genetic variation is associated with gene expression differences in these 2 cell types. These are known as expression quantitative trait loci, or EQTLs. EQTL data can be plotted just as any other association data, and the 2 different cell type EQTLs are shown here in these 2 tracks. I'm focusing on just one of the EQTLs here to the gene CENPW. Notice that the same locus that was associated with cortical surface area that I showed previously is also associated with expression of CENPW in progenitor cells, but not in neurons. So what these data allow us to do is to move from a non-coding genomic loci to a gene of interest, in this case, CENPW. Next, we used our chromatin accessibility data to identify where genetic variation impacts promoter or enhancer activity. These are called chromatin accessibility QTLs, or CAQTLs. Here we show those again for both progenitors and neurons relative to a chromatin accessibility peak right at the promoter of CENPW. Again, we show that the same locus that's associated with cortical surface area and expression of CENPW is also associated with gene regulation at this chromatin accessibility peak, this time in both progenitors and neurons. This shows that a genetic variant impacts non-coding gene regulatory activity as well as gene expression. If we zoom in further in the peak, right at the promoter of CENPW, we can focus on the SNP within the peak as most likely to be causal. It's most likely to be causal because it is in a functional genomic element, and in this way we can suggest a causal variant among all of the associated variants that are in high LD. At the top here, you can see coverage plots for chromatin accessibility based on each of the 3 different genotypes. This is the average chromatin accessibility for donors of these genotypes. Using the combination of this data, we have identified an individual SNP within the locus that impacts regulatory element activity and gene expression of CENPW that also influences cortical surface area. So once we have identified an individual putatively causal SNP, we can determine whether that SNP impacts transcription factor binding motifs. These are sequences in the genome where transcription factors preferentially bind and are shown here. This SNP disrupts the motif of 4 transcription factors, ATF1, ATF2, ATF4, and CREM. The T allele of this SNP preserves the motif, whereas the C allele breaks the motif. This means that each of these transcription factors is more likely to bind to the T allele than it is to the C allele, which we hypothesize then has downstream impacts on CENPW expression. This suggests a regulatory mechanism by which a single base pair change in the genome can lead to alterations in gene expression and downstream impacts on brain size. Interestingly, this locus is not only associated with cortical surface area, but is also associated with educational attainment. Again, another colocalization. So this locus is not just impacting brain structure, it's also impacting brain function. By integrating this multi-omic GWAS data, we can begin to discover mechanisms by which non-coding genetic variation impacts brain structure and cognition. At one locus that I've highlighted here, we can propose a model whereby a single variant impacts transcription factor binding in human neuroprogenitor cells during development. The C allele at this variant has disrupted binding of 1 or more of 4 transcription factors. This leads to decreases in CENPW expression, which presumably impacts mitosis in these progenitors. Then, in adulthood, cortical structure and cognitive ability are affected as well. Interestingly, CENPW forms a centromeric protein that functions in mitosis, and mutations in other centromeric proteins like CENPJ are known to cause microcephaly and severe intellectual disability. There are several experiments that we'll be working on in the lab in the future to determine if these transcription factors are indeed impacted by this SNP. Those are allele-specific chromatin immunoprecipitation assays. Using these same data in the future may help explain regulatory mechanisms underlying risk for neuropsychiatric disorders as well. So I'd like to thank you all for listening to this online talk. And I would especially like to thank the members of my lab who completed this study. I'd like to thank Nil Aigun, she's here, and Dan Liang, she's here, who did both the EQTL and CAQTL work. And I'd also like to thank Angela Elwell, who grew all of these cells, cultured all the cells, and also performed all the library preparations for the study. So thanks very much, everyone.
So today I'm going to tell you about molecular cellular diversity in the neocortex across different human cortical areas and between human, non-human primate, and mouse. So studies for many decades have shown diversity across different regions of the cortex in their connectivity, in their cytoarchitecture, and in gene expression differences, particularly early on based on bulk RNA-seq data and microarray data. And those expression differences can vary as a gradient across the anterior to posterior axis or medial lateral axis, but also show discrete transitions, for example, in some of the primary sensory areas. So we've been taking a single-cell transcriptomics approach to understanding cellular diversity, as have many other groups. And this is really a powerful approach because it's high throughput. It defines thousands of features within individual cells. And it really allows one to, in a relatively unbiased way, define the cellular diversity within a tissue. And that can be extended to many species and many different tissues. And finally, with that data in hand, it also defines the molecular profile of cells that can form hypotheses about cell function and also define markers that allow genetic targeting of cell types. So again, what we do is first is take a single cell survey of a region, perform unbiased or unsupervised clustering to define cell types within that region. And then to understand the spatial distribution of those types, we can either use the initial dissection information, but even better is to then label these cell types using marker genes in tissue, using a variety of approaches. And finally, developing viral tools and in model organisms, transgenic models, to profile other cellular features. So today, I'll talk about the first part, and in particular focus on cross-species variation in primary motor cortex, and also cross-areal variation within the human cortex. So in order to profile individual cells from human cortex and other human brain regions, we focus on postmortem tissue where we can select the regions of interest. And this involves frozen, coronally slabbed tissue where we can dissociate the individual nuclei, often labeled with nu-n, and sort to enrich for neurons. And finally, profile with one of two technologies mainly, the SmartSeq version 4, full-length transcript information, and then the higher throughput droplet-based method from 10x Genomics. So we initially started the survey in middle temporal gyrus because that's a region where we often can receive neurosurgical acute sliced tissue from epilepsy resection surgeries, and allows profiling of other cellular features. And so in this region, we profiled almost 16,000 single nuclei with SmartSeq and with very high gene detection, and found a number of very discrete clusters based on their RNA profiles that were shared across donors and had a laminar structure. So we could arrange these clusters into a tree based on their shared transcriptomic profiles, and found inhibitory types are distinct from excitatory types, and all the glial types are quite distinct from each other. And within the inhibitory types, we see the two broad classes of types that derive from different progenitor populations, the two ganglionic eminences, two of the ganglionic eminences. Moreover, we see many of these types are quite rare, although some are common. And many types, including both excitatory and inhibitory neurons, are quite restricted to one or two layers. So more recently, we've taken a comparative approach to look at a different cortical region, M1, that is more clearly functionally conserved across species. And this was in collaboration with a number of labs through the Brain Initiative Cell Census Network, where we profiled not only RNA-seq data with SmartSeq and 10x Genomics, but also DNA methylation and open chromatin. And with this approach, we took the same approach as in MTG, where we defined taxonomies of cell types within each species and identified approximately 100 types within each species. We could then leverage co-expression, shared co-expression that's been conserved, to integrate these nuclei into a common space and then perform unsupervised clustering to match types between the species. And in fact, we found matches between virtually all of these types, some of which were one-to-one and some many-to-many. Moreover, we had the epigenetic information as well, and we could perform an integration between both the DNA methylation signatures and the open chromatin signatures and the RNA clusters. So here on the left, I'm showing clusters that were defined in an unsupervised way based on DNA hypomethylation of different genes. And those could be mapped based on the inferred expression of marker genes to the RNA clusters, either one-to-one or one-to-many. And there really was a sort of striking conservation of these gene regulatory signatures and expression. That we performed a similar comparison between the TAC open chromatin data and RNA. And with these epigenetic data in hand, we could ask questions such as, what are the developmental origins of cell types? Are there conserved transcription factors that may be responsible for defining cell type identity? And so here I'm showing on the left, in the middle panel, the LAMP5 subclass of interneuron. And here we looked at both the RNA expression of a number of transcription factors and also motif enrichment in DNA hypomethylated regions and open chromatin, and found a very consistent signature across those modalities and strikingly across species. Moreover, each of these subclasses was distinct from the others, shown on the TSNA production on the right, where different colors represent different neuronal subclasses and different shapes represent the different species that are intermixed within those subclasses. While there was conservation, we also saw distinctions between the species. One of the more striking ones was in the cell type proportions of, in particular, excitatory neurons versus inhibitory neurons, where the excitatory-inhibitory balance is much higher in mouse than it is in human. And within the excitatory neurons, we see an expansion of the layer 2-3 layer 2-3 intratelencephalic neurons, which corresponds to the relative expansion of those layers in primates. Moreover, expression patterns showed some conserved markers, but also many species-specific markers. And within the same gene, up to 20% of isoforms showed differential usage between human and mouse that was even more pronounced in non-neuronal cells than in the inhibitory or excitatory neurons. With these data in hand, we could also compare the human M1 data set to our original MTG taxonomy and try to understand to what degree cell types are conserved between two cortical regions. In particular, we could ask, even though there isn't a distinct layer 4 based on cytoarchitecture within M1, perhaps there are molecular signatures of layer 4-like cells in M1. And this would be important to know because different layers receive different inputs. The layer 4 receives a strong thalamic input and implies something about the connectivity of those cells if they look like a layer 4 type. So this has been described many years ago by Cahal and his contemporary Brodmann, where Cahal argued that there was a layer 4 in the adult human cortex, while in Brodmann argued that that was only present in fetal development and disappeared. So we took a similar strategy as we did between species to integrate, based on shared co-expression, the excitatory neurons of M1 and MTG, and found good mixing between these two sets of nuclei, shown in the different colors in this TSNE plot on the left. And all of the major subclasses of excitatory neurons are represented. In particular, if we zoom in on the layer 4-like types, there are 4 different types in MTG and only 2 present in M1 that match 1-to-1 to these types in MTG. So there is a lower diversity of these types in M1, but there are some present. In particular, this one type is highly enriched in layer 4 based on in situ labeling and has a more scattered and sparse distribution in M1. So, one hypothesis is that these cells really will have a morphology that resembles a layer 4-like type, and that's pending new data. So we can really extend this approach of comparing between areas to many different areas in cortex, and we focused on 6 initially. These include the two areas that I mentioned, in addition to somatosensory cortex, primary auditory cortex, visual cortex, and anterior cingulate. And we derived these from, again, postmortem tissue from 3 human donors, and this analysis focused on SmartSeq data from those different regions. We defined a taxonomy of types, pooling all of the data together and performing unsupervised clustering. And we see a similar trend as an individual region where many types are rare with a few common types. And then we can ask for each of those types, how is it present in all the different regions? And for non-neuronal types, this is the case. So we see all of these types in all regions. And that's also true for the inhibitory neurons, with the exception of this one parvalbumin positive interneuron that's enriched in the primary sensory areas. In contrast, the excitatory neurons show much more diversity across regions. In particular, the layer 4 enriched types are more diverse in the primary visual cortex and in primary sensory areas in general. and also these deep projecting layer 5 subcerebral types, which represent only a few percent of all the layer 5 excitatory neurons, but can have been described in the literature to have a very diverse and distinct morphologies, and in non-human primate have been shown to have distinct projection patterns. So for example, these include the cortical spinal projecting large BET cells in primary motor cortex. So I've shown that there are on the order of 100 cell types in each cortical region with distinct markers and layer distributions. Excitatory neurons vary more across areas than other cells. And moreover, cortical cell types have largely been conserved, at least at a coarse level, for over 65 million years, and therefore likely have functional significance. Developmental mechanisms are suggested for cell type identity based on having a conserved transcription factor code. And finally, there are really distinct species specializations for both cell type proportions and expression that may point to different adaptations for ecological niches and will be interesting to explore in future work. And all of these data are available through this portal on our website and also through the NEMO data archive. And I'd like to thank not only the BICCN collaborators who contributed to this work, but also all the people at the Allen Institute and our founder, Paul Allen, for his vision and support. Thanks for your attention, and I'm happy to take any questions.